Hello students and a warm welcome. Myself Dr. Tripti Malik Ahuja, your educator. This is another video for the preparation of CUET examination for admission to MSc Microbiology, MSc Biotechnology and MSc Life Sciences. This is actually the question paper of ICMR JRF conducted in July 2019. There is a previous video in which first 50 questions were discussed. You will find the link in the description box below and in the I button here. In this video you will get the next 49 questions and there will be another video, the next video, in which last 51 questions will be discussed. This next part, the part 3, you will also get very soon. If you are new to this channel and you have not subscribed it, then subscribe it and press the bell icon to get the notifications of the new videos. So now let's get started. This is the part 2, section P. The questions are of life science. In the part 1, there was section A. The questions were from the aptitude. Let's get started. These first questions are from the biochemistry. The question number 51. The highly repetitive DNA in the eukaryotes occupies the which fraction of the CUET curve? The options are 1 slow, 2 intermediate, 3 fast, 4 all of the above. Out of this, the correct option is the option which is the number 3, that means fast. CUET analysis is based on the principle of DNA reassociation kinetics. When there is more repetition, there is faster reassociation. Students, I am here reading the question and I am also telling you the options, out of which the option you think is correct, you can note it down on a paper. And I will also tell you the correct option. If your answer does not matches with the answer what I am telling you, then you can think it over, and still if you are in any doubt, you need an explanation, you can write it in the comment section. I will give the reply to each and every comment. So let's get continued with the next question. Question 52 says, which of the following type of enzyme inhibition is also called as end product inhibition? Options: 1 substrate regulation, 2 feedback inhibition, 3 competitive inhibition, 4 non-competitive inhibition. The correct one is 2 feedback inhibition. The inhibition of enzyme hexokinase by glucose 6-phosphate in glycolysis is an example of the end product inhibition. Question number 53. Cholesterol does not act as precursor for 1 cardiolipin, 2 progesterone, 3 cortisol, 4 estradiol. Here the correct one is the cardiolipin. Cardiolipin is a phospholipid found exclusively in mitochondria and its precursor is cytidine diphosphate diacylglycerol. Next question. Allopurinol is used for the treatment of gout. It is an inhibitor of 1 thymidylate synthase, 2 xanthine oxidase, 3 hypoxanthine guanine phosphoriboside transferase, 4 adenosine phosphoriboside transferase. The correct answer is the option number 2, xanthine oxidase. Xanthine oxidase is the enzyme which converts the purine nucleotides to uric acid. Hence, it can be used for the treatment. If there is an inhibitor of xanthine oxidase, that can be used for the treatment of gout. Gout is because of the excessive production of uric acid. Question number 55. Alpha oxidation of fatty acids takes place in, options are 1. Endoplasmic reticulum, 2. Cytosol, 3. Mitochondria, 4. Peroxisomes. The correct answer is the number 4 option, which is peroxisomes. Question number 56. Which of the following enzyme participates in both the citric acid cycle and the electron transport chain? Option 1. NADH dehydrogenase. Option 2. Malate dehydrogenase. Option 3. Succinate dehydrogenase. The option 4 is isocitrate dehydrogenase. The correct one is the option number 3, succinate dehydrogenase. It transfers the electrons to ubiquinone UQ in the ETS. Question number 57. Which of the following molecule yields maximum number of ATPs upon oxidation? Options are 1. Glutamate, 2. Pyruvate, 3. Palmitate, 4. Glucose. This is a very simple question. Hope you have guessed the correct answer. The correct answer is 3. Palmitate. Next question, question number 58. Which is true about enzymes? All enzymes are protein. All enzymes are vitamins. All enzymes are not proteins. All proteins are enzymes. This is a tricky question and the answer is the option 3. All enzymes are not proteins. Some of you must have said all enzymes are proteins. It could be true, but there is one exception, which is the ribozymes. So ribozymes are not the protein. Actually, these are the RNA sequences. So the correct one is the all enzymes are not proteins. Question number 59. Wavelength range of absorption peptide bond is 190 to 230 nanometer, 240 to 270 nanometer. Option 3, 160 to 180 nanometer. Option 4, 250 to 280 nanometer. Here the correct one is the option 1, which is 190 to 230 nanometer. Question number 60. Pick up the amino acid which is present in the body but not found in proteins. Option 1, arginine. Option number 2, 4-hydroxyproline. Option 3, ornithine. Option 4, selenocysteine. The correct one is the option number 3, which is ornithine. Ornithine is produced in liver during the formation of urea and it is not found in the proteins as such. Now next questions are from cell biology. The question number 61. ALU elements in human genome represent option 1, exons, option 2, introns, option 3, nucleotide repeats, option 4, transposable elements. The correct one is the option number 4, transposable elements. Actually, the ALU elements, these are retrotransposons and they cause the transcription of nearby genes. Question number 62, a very simple question. Hope you will guess the correct answer. The question number 62 says, which of the following structures is known to maintain the shape of a cell? Option 1, ribosomes. Option 2, microtubules. Number 3, nucleus. Number 4, mitochondria. So the correct answer is 2, microtubules. Microtubules are also called cytoskeleton of cell. Question number 63. Calmodulin contributes to signal transduction by binding to 1, CAMP, CMP, 2, calcium, 3, magnesium, 4, sodium. The correct one is the 2, calcium. Question number 64. Another easy question, very direct question also. Crossing over occurs in which phase? The number 1 prophase 1, number 2 telophase 1, number 3 anaphase 1, number 4 metaphase 1. So you have got the correct answer, I know, and the correct answer is the option 1, which is the prophase 1. I hope you are enjoying these questions. Question number 65 says, the non-sister chromatids twist around and exchange segments with each other during option 1 lactotine, option 2 dikinesis, option 3 diplotin, option 4 is pegitine. The correct one here is the option 4, which is pegitine. Question number 66. Human genome contains about 2 billion base pairs, 3 billion base pairs, 4 billion base pairs, 5 billion base pairs. Note all the options are in billions. So it is important to remember the correct figure, and the correct one is the option number 2, which is the 3 billion base pairs. Question number 67. Which of the following amino acid is present abundantly in histones? The options are 1 aspartic acid, 2 tryptophan, 3 arginine, 4 glycine. The correct one is the option number 3, which is arginine. So these were the questions from Mol Bio. Now the next section is of the molecular biology. Question number 68. Which of the following growth media would you expect to result in synthesis of high levels of messenger RNA for the enzymes of the E. coli lactopron? The option number 1 high glucose, high lactose. Number 2 low glucose, low lactose. Number 3 high glucose, low lactose. Number 4 no glucose, high lactose. So think it over, as all the options, it is important to exclude the options here. The options, they are quite similar, they are somewhat differing also. So you have to see how these options are different and what will lead to the E. coli operon, which is usually switched off when it switches on in the condition when there is no or lesser glucose and there are other substrates other than the glucose. So here the most appropriate option is the no glucose, high lactose. So if there is no glucose and there is higher amount of lactose, then the lac operon will be switched on and the high levels of the enzymes will be produced. Question number 69. What is the mode of action of exonuclease 3? The options 1 is exonuclease 3 acts on single stranded DNA in 3' to 5' direction. Option 2, exonuclease 3 acts on double-stranded DNA in 5' to 3' direction. Number 3 option, exonuclease 3 acts on single-stranded DNA in 5' to 3' direction. The number 4 option, exonuclease 3 acts on double-stranded DNA in 3' to 5' direction. First of all, you have to think whether the strand on which this enzyme acts is single-stranded or double-stranded. So there are 2 options of single-stranded, there are 2 options of double-stranded. So exclude out the options which are double-stranded, obviously. Now tell me fast in the comment section what would be the correct answer. So the correct answer is the option number 69, because exonuclease 3 acts on single-stranded DNA and the direction is 3' to 5' direction. So that was a tricky question. Now the question number 17, the specific DNA sequences to which the transcription factors bind are referred to as number 1 replication elements, number 2 blocking factor, number 3 transcription factors, number 4 regulatory elements. This is a direct question and the answer I have answered is here the number 4 regulatory elements. Question number 71. Which of the following statement is incorrect regarding DNA methylation? Option 1. S-adenosylmethionine SAM is one of the most important methyl donors. Number 2 option, it is catalyzed by enzymes. Number 3, occurs at CPG islands. Number 4, mainly G is methylated. You have to tell this statement which is incorrect. So here the option which is correct is the fourth one. It says mainly G is methylated. Actually it is the incorrect statement. In addition to G, C is also methylated. So this is the incorrect statement. Question number 72. Which of the following is not an example of post-translation modification? Another question, tricky one. It is saying not an example. The options are option 1, addition of prosthetic groups, option 2, proteolytic processing, option 3, mRNA splicing, option 4 is the loss of signal sequences. The correct one is the option 3, which is the mRNA messenger RNA splicing. Question number 73. Which of the following statement is incorrect about the gelatin coat? The statement 1 says a codon is a triplet of nucleotides that code for a specific amino acid. Option number 2 says a specific first codon in the sequence establishes the reading frame. Option 3, a codon specifies more than one amino acid. Option 4, nucleotide triplets are read in successive non-overlapping fashion. So here the option which is the option which is actually the incorrect statement, that is the option number 3. The option number 3 says a codon specifies more than one amino acid. This is incorrect. A codon specifies for only one amino acid. Question number 74. Human telomers consist of tandem repeats of sequence. The sequences are number 1 TTA, GGG, the N number of the monomers. Next the TT, double A, triple G. Third is the double T, double A, double G. The fourth one is the, actually it is written 1, it should be T, double T, triple A and double G. So here the correct one is the first sequence, which is the double T, single A and then the triple G. You have to remember it. Question number 75. The original codon changes to stop codon in which type of the mutation? Options are 1, sense mutation, number 2, missense mutation, number 3, nonsense mutation, number 4 is the reverse mutation. So here the correct one is the option number 3, which is the nonsense mutation. Question 76 says, if the amount of G in a DNA sample is 20%, what will be the amount of T? The options are 140%, 250%, 330%, 420%. You have to find it out, calculate it. I am giving you few seconds and write the answer in the comment section. I hope you have done it. The correct one is 30%. So I'm telling you once here, and if you need more explanation, you can write in the comment section. I will explain you again also. When there is G, which is 20%, so C is also 20%. So 20 G20C makes 40%. 100 minus 40 will be the A and T. So 100 minus 40, that is 60. So out of 60, the half of 60 is A and the half of 60 is T. So half of 60 is 30. So T is 30 and A is also 30. Now moving to the next question, the question number 77. Which of the following takes place in both bacterial as well as eukaryotic messenger RNA synthesis? Takes place in both. That means common in both. The options are 1 poly A tailing, second option is 5' capping, third option is splicing, fourth option is DNA dependent RNA synthesis. So here the correct option is the option number 4, DNA dependent RNA synthesis. Question number 78 says, Tata box number option 1 is present on the template strand. Option 2 says present about 70 base pairs away from the transcription start site. Option 3 serve as a signal for attachment of RNAP2. Option 4 acts as a silencer of the G. The correct one is the option which is the option 2, present about 70 base pairs away from transcription start site. The Tata box is present on the coding strand where the RNA polymerase enzyme binds. So these were the questions from the molecular biology. Now next are the questions from the subject immunology. The question number 79. Which of the following substances will not stimulate an immune response unless they are bound to a larger molecule? Option 1 hapten, option 2 antigen, option 3 antibody, option 4 is virus. The correct one is the option 1, which are the hapten. Haptens are non-immunogenic molecules. Question number 80 says, which of the following is the major immunoglobulin in human serum, accounting for 80% of the immunoglobulin pool? The options are IgM, IgE, IgD, and the fourth option is IgG. The correct one is the option 4th, which is the IgG. Question number 81. Type 1 hypersensitivity is mediated by which of the following immunoglobulins? The option 1 is IgA, option 2 is IgG, option 3 is IgE, option 4 is IgM. Out of this, the correct option is the option 3, which is IgE. Question number 82. Acute inflammation characteristically involves option 1 influx of mast cell, option 2 capillary endothelial cell enlargement, option 3 influx of neutrophils, option 4 is influx of macrophages. The correct one is the option 3, which is the influx of neutrophils. Again, if you want an explanation of any of the correct option or you think another option should be correct, you can write it in the comments. Now question number 83. A tissue graft between two people who are not genetically identical is termed as, number 1 is isograft, number 2 is heterograft, number 3 is xenograft, number 4 is allograft. So the option here is the number 4, which is the allograft. Question number 84. Cell-mediated immunity, number 1, can be transferred passively using serum. Number 2 is mediated by B and T cells. 3 is mediated by T cells, macrophages and interleukins. Number 4 forms the major part of the innate immunity. So out of this, the correct one is the option 3, mediated by T cells, macrophages and interleukins. Question number 85. B cells differentiate to form Number 1 option is plasma cells, number 2 effector cells, number 3 plasma cells and memory cells, number 4 germinal cells. A very easy question, and the correct answer is the option 3 plasma cells and the memory cells. Question number 86. Opsonin is the number 1 cell wall component, number 2 plasma component, number 3 serum component, number 4 is cytoplasm component. The correct option is the option 3, which is the serum component. Question number 87. The class of immunoglobulin, number 1 option is determined by class 1 and class 2 major histocompatibility complex proteins. Option number 2 is determined by the carbohydrate attached to the light chain. Number 3 determined by the antigen. Number 4 determined by the heavy chain type. The correct option is the option 4. It is determined by the heavy chain type. Question number 88. J chain is present in which antibody? The option 1 is IgG, option 2 IgM, option 3 is IgE, the option 4 is IgT. Out of this, one antibody has the J chain, and I think you have written it correctly. Yes, the answer is the option 2, which is the IgM. IgM is the antibody which is the produced usually in the secondary immune response, and this is a pentameric antibody. The monomers are joined by the J chain. In addition to the IgM, the IgA is also a dimer which is also having a chain uniting the two monomers. These were the questions from immunology. The next subject is the physiology. Question number 89. Which of the following structures is an example of lymphatic vessel? The options are 1 thoracic duct, the option 2 parotid duct, option 3 is the bile duct, option 4 is the cystic duct. Out of this, the correct one is the thoracic duct. Question number 90. Which connective tissue cells are responsible for synthesizing collagen fibers? The options are 1 macrophages, 2 fibroblasts, 3 mast cells, 4 adipocytes. Here the correct answer is the option 2, which is the fibroblast. Now question number 91. Structure in descending order related to bile duct. Number 1 is head of pancreas, first part of duodenum, lesser omentum. Number 2, first part of duodenum, lesser omentum, head of pancreas. Number 3, lesser omentum, first part of duodenum, head of pancreas. The option 4 is head of pancreas, lesser omentum, first part of duodenum. Here the correct one is the option number 3, which is the lesser omentum, first part of duodenum, head of pancreas. Lesser omentum is a double layered sheet of peritoneum. Question number 92. Coronary arteries arise from 1 ascending aorta, 2 arch of aorta, 3 subclavian artery, 4 descending aorta. The correct one is ascending aorta. Question number 93. Thyroid hormones act through number 1 nuclear receptors, number 2 plasma membrane receptors, number 3 is cytosolic receptors, number 4 ER receptors. The correct option is the option 1, which are the nuclear receptors. Question number 94. Which of the following causes bicyanosis? The options are 1 cotton dust, 2 benzopyrene, 3 peroxyacetyl nitrate. Here the correct option is the option which is the cotton dust. So it is seen in the individuals who are working in the textile industry. Question number 95. Which part of the body best represents the core body temperature? The options are oral cavity, number 2 axilla, number 3 rectum, number 4 is the nasal cavity. So here the correct option is the option number 3, which is rectum. Question number 96. The velocity of blood flow is highest in number 1 ascending aorta, number 2 capillaries, number 3 large veins, number 4 is pulmonary trunk. The correct one is the option which is the option first, which is the ascending aorta. Question number 97. Insulin increases entry of glucose in the liver cells by increasing the number of glucose transporters GLUT4 on the hepatocytes. The statement 2 is activity of glucokinase, which decreases intracellular free glucose, thus promoting diffusion. Third one is activity of sodium potassium ATPase, which utilizes glucose for its energy requirements, thus decreasing free glucose within the cell. Number 4, activity of transcription factors for the production of glucose transporter in the hepatocytes. So here the correct one is the first, which is the number of glucose transporters GLUT4 on the hepatocytes. Question number 98. Physiological dead space is calculated by 1. Dalton's law, 2. Bohr equation, 3. Boyle's law, 4 is the Charles law. The correct one is the option 2, which is the Bohr equation. So these were the questions from the question number 50 to the question number 98. Soon the next video, you will get the next video on the remaining questions, the question number 99, which are from genetics subject, from zoology, from botany, and from microbiology and biotechnology, bioinformatics and biophysics and environmental science, and some are from the veterinary science also. So stay tuned. All the best. Take care. See you in the next video. Bye for now.